Hello and welcome to our Lakeside chat. We are David Wofford and Katie Noonan, co-chairs of the Rotary Nature Center Friends. And we're so happy that you can join us tonight.
As we begin our program, it is important for us to acknowledge that we are on stolen land, that Lake Merritt is part of Ohlone territory. We hear now from Corinna Gould, spokesperson for the Confederated Villages of LaShawn and co-founder of Segurate Land Trust.
Good afternoon, relatives. My name is Corinna Gould. I am the spokesperson for the Confederated Villages of LaShawn. We are here today at what most folks think of as Lake Merritt. We are in the territory of Huichin. Huichin is actually a territory that encompasses 6 Bay Area cities: Oakland, Berkeley, Alameda, Emeryville, Albany, and Piedmont. And this was a place of abundance. I'm so happy that people from all walks of life that now come into our territory can enjoy this beautiful place that my ancestors have enjoyed since the beginning of time. My relationship to the land that I have been born to, raising my children and grandchildren here, has been to tell the story, the truth of what happened on this land before other people came here. I'm hoping that as we begin to learn these lessons of fires in California, the pandemic that's happening, that human beings come back to living in reciprocity with the earth.
Thank you, Corinna, for an inspiring message and something that is so meaningful today. We're very happy to be here tonight to learn more about the reciprocity and interaction and the complexity of our world and of our of the earth. And so I would like to welcome everybody again and say thank you for returning, those of you who are returning, and for those of you who are new, welcome for joining us. We're going to take a moment to tell you just a little bit about ourselves. So we are Rotary Nature Center Friends, an independent, all-volunteer 501c3 nonprofit since 2021, providing online and in-person programming for all the people of Oakland. We engage the community in real science and stewardship activities, bringing people and nature together in the heart of downtown Oakland to steward the nation's first wildlife refuge, the Lake Merritt Wildlife Refuge. Here's a list of the focus of some of our activities, both in community science and environmental justice. And I won't read all of the entire all the sentences, but we're very active in water testing and also in monitoring the plankton diversity in the lake, and also through iNaturalist and help with the California Academy of Sciences and other local naturalists, just monitoring biodiversity. We involve young people and reach out to all generations living in Oakland with our programs to build community and to make people aware of some of the issues and opportunities that exist in our community. We've pulled together a board of directors with many different talents and interests, and we are poised, ready to do whatever we can to further our our mission of bringing people and nature together and creating an interpretive science community and stewardship community at Lake Merritt. And so I'm going to stop my screen right here because I want to say how happy we are to have you here And to mention just a few of some of the things that we've been doing in the past month, we've spent quite a bit of time on water testing as the year comes to an end, and we're taking the most detailed look at the water quality or the water characteristics of Lake Merritt than has ever been done before. We're working with the city of Oakland and also the water boards, and collecting a lot of data that will and give us some baseline going forward in stewarding the lake. We've had a number of different young youth groups come out, young little kids and high school kids. We have water justice fellows from the New Voices Are Rising program with the Rose Foundation, and we've had some involvement with the California Climate Action Corps fellows with the city of Oakland, coming out to help us test water, steward our salt marsh that we have, our little demonstration salt marsh. And now we have just had a influx of fish that are from the ocean and from the bay. We have had an influx of Chinook salmon coming into the lake, and are involving local community observers and young people in analyzing and finding and taking down information about that. So a lot of very interesting things happening here at Lake Merritt, and I want to thank everybody involved in helping us to do so. One of the things that we did is working with the California Academy of Sciences. We've been working with Snapshot Cal Coast, and also the City Nature Challenge, which I think Leslie, who's our guest, may have been one of the founding people to start that contest between LA and I got it wrong. Well, anyway, it was the LA County and the California Academy that set up those programs, and we've done a number of them this year. So I'm going to finish with that. We would love to share a lot of photos, but we don't have time. And I encourage you to go to our Facebook site and our website to see some of the things that we've been doing. So without too much further ado, I think we're ready to introduce our special guest tonight, Leslie Harris. So Leslie is a Polychaete specialist, and she is currently the senior Polychaete collections manager at the LA County Museum. And I'm going to, I've learned a couple of interesting facts about her. Has a huge bio, and with many interesting accomplishments and interests, but among them that she has 24 godchildren, human, and she also has named a number of different species, and her name is part of the genus name of, or the species name of these species. So I believe she said there were 5, and one of them is a shrimp that was named after her in the genus level. And then there's a bubble shell snail, which is a kind of snail that we see a lot in Lake Merritt. This is in the genus Feline, and it has the species name Harrisii. And then she has a number of other species with carrying her name, which are polychaetes. So I think that I would ask Leslie if she could tell us a little bit more about herself and how she got into this career and fascination with invertebrates and polychaetes in particular.
Well, like so many people, I fell into my career. I started volunteering while in college, not volunteering, I was actually paid part-time at a marine monitoring agency, a research institute. And I was doing computer coding and whatever minion tasks they needed. And this was back in the days when you coded by printing things out on sheets of paper with alternating green and white squares. And then you took that over to a computer center and they did punch cards. So yeah, that was fun. And then one day someone came into our room where the minion sat and said, "We're starting a benthic lab and you're our new taxonomist." They brought somebody in to train us. I learned everything to start, but for whatever reason, I was especially good at polychaetes and I found them fascinating. And that was it. And I've been working with them ever since.
That's wonderful. So many, it's really hard to figure out, what your career goal is. And then sometimes you just fall into it and it becomes something so satisfying and such a wonderful, enriching interest as well as a career. So that's a really great story. So we're so happy to have you here tonight because we have a lot of polychaetes in Lake Merritt, but we're not aware of them because we seldom see them. And some of them are pretty tiny. And so, and then there's some lingering questions out there that I think it's fascinating that this is all not set in stone, that there's a lot of research done to figure out more about the polychaetes in Lake Merritt and adjacent areas. So I'm really looking forward to what you have to tell us. And let's see, are we ready to go to share screen?
Yeah, absolutely. And I just want to say that for the most part, this is basically going to be a parade of images, mostly taken from iNaturalist. So some of you may recognize your photos as the slides go by. Some of my own, and with a surprise contribution by one of our very eminent members here. And yeah, it's a rogues gallery of this is who you've got in the lake. So let's get started on this. Okay, well, this came about because I escorted a pair of Australian colleagues up to San Francisco. And in the course of that, we met up with Robin, a nudibranch mom, and Katie and some other people at Lake Merritt. And of course, I wanted to see all the polychaetes there. And Katie and I got to talking and she said, "Oh, it'd be so great if you could talk." And I said, "Sure, why not? I'm always happy to spread the love of polychaetes to an unwitting world." So I actually don't know that much about Lake Merritt, just a broad outline. Fascinating place with its history and how it's changed over the years. I do want to say that our special contribution is by Dr. Carlton. Jim, just the other day, surprised me with an email and said, "Here's a history of polychaetes in Lake Merritt." And, which was delightful. Certainly nothing that I had pulled together. One thing I want to emphasize at the start, that polychaetes and oligochaetes are all annelids, along with leeches and cypunks and echuroids now. And I just want to emphasize that most people know oligochaetes, and to me, they are totally boring, because no matter where they are in the world, whether they're a few millimeters long or a couple of meters long and an inch wide, they pretty much all look alike, reproduce alike, feed alike, same mode of life for the most part. polychaetes are not boring. They come in a vast variety of shapes, behaviors, reproduction. I mean, they do everything, every mode of asexual that you can imagine, and of course, sexual reproduction, hermaphrodites, sequential hermaphrodites, simultaneous hermaphrodites. Some alternate sexual and asexual reproduction. Some spend their whole lives swimming like fish. Some are big enough to catch fish for their food. So really fascinating animals. So thanks to Jim, a partial history of Polychaete collections. So the earliest known collection is everybody's favorite Australian tubeworm, Phycopomatus enigmaticus. Jim gave a wonderful talk on this a couple of years ago. I thoroughly enjoyed it when he did. Now, as he points out, that polychaetes were known and even collected in the 1890s. So people have been looking at them for a while, but not much has been actually published about them. So we don't have many lists. In the 1930s, Saul Light, who is at University of California at Berkeley, and his graduate student, Olga Hartman, were collecting polychaetes in the lake. And a lot of these polychaetes ended up at my museum when University at Berkeley decided to get rid of them during World War II. And he passed them on to Olga, who at that time was working at the Allen Hancock Foundation on the USC campus. And that collection was eventually given to my museum. I was working with the collection at USC at the time, and they basically just gave me to the museum along with the collection to take care of it. So in 1938, in this wonderful laboratory syllabus, the genesis of the Light and Smith manual, as we know it today, were these 7 species. On the left are the names that were used in that publication, and on the right are the names that we know today. Dr. Light expanded that into the first published Light's manual in 1941, and had an actual key to the annelids of Lake Merritt, which was done by Olga Hartman, who became one of the world's leading experts on polychaetes. And just a few species, she added a few more. So 1, 2, 3, 4, 5, 6, 7, 8 species. Not very much. I found this little bit from Jim really fascinating about his personal collection to someone who was working in Lake Merritt in the 1950s. And I think for you, there's this connection to the park itself. Jim, would you like to talk a little bit about this? Because I can only read your words, but you lived it.
Yeah, and this was my father was interested, of course, in everything around the lake, not just the birds, but everything else around the lake. And so certainly there were a lot of times when there was a lot of mud flats that were exposed and so on, which were good habitats for some of these polychaetes. And so there's just a growing curiosity about the inverts there, including the polychaetes. And so my dad was always interested in getting some real scientists in there to look at some of these invertebrates. And of course, when Jim Carlton came along, he really started to dig into those things. But that kind of started this deeper dive into invertebrates in the lake, and which is, there's been a number of scientists through the decades that have contributed to that, as you've cited.
Yeah. And I'm really tickled by what he said about little question that had they remained at the Rotary Nature Center during the reconstruction and renovation, they would have been discarded. So thanks to your father giving the specimens to Jim, they've been preserved and are still available for study. So after that, well, then came Jim Carlton himself with his, well, being with his intrigue with the coral reef that was living in there, as it was known then. I've never been able to say this name right, Marcelliella enigmaticus. So I know Jim was saddened when the name was changed to Phycopomatus, but it's so much easier to say. Really quite happy about that. So in his dissertation, he included this list of worms, and it's still a fairly short list: 1, 2, 3, 4, 5, 6, 7, 8 polychaetes, which is not much at all. And the fact that there are so few tells us that the area remained relatively unchanged over the years. So some people, were doing collections in there. Not much has been written up. I love this one that in 2016, Jim and Andy Chang from CERC did the Carlton plus 50 extensive sampling. I love these long-term surveys that we can see how things have changed, or in this case, stay the same. And again, just 7 species of polychaetes, which actually tells me that the sampling was not very intensive, or that the area that was sampled was small and the same type of habitat, because there are certainly more species than this. So if we go to iNaturalist and type in Lake Merritt and Polychaete, we get these species here. So 18. We take just a little closer look, we can immediately eliminate this one. Over the last couple of days, I looked at all 410 observations of polychaetes recorded from Lake Merritt. And this serpula, it's actually a fungus. Somebody just hit the wrong name, because there is the fungal genus serpula and there is a polychaete genus serpula, and people get it wrong all the time. So we can eliminate that one. So now we're down to 17. But looking at a variety of records, my own collecting, and a sample that Katie sent to me in October, it's more like 30. And then I added 3 in that are likely to be at least in the more saline portions. But yeah, Capitella, Nodomastus, at least 2 species of Ceratulids. In the sample that Katie sent me, they were small and torn, so I really couldn't identify them. Seriformia, Ophiotrocha was in the sample. Marphysa californica, Marphysa sanguinea has been reported. That's probably an incorrect identification. 4 species of Nerids, and I'll get into all of these later. 3 phyllodocids, 2 scale worms, 4, possibly 5 different fan worms, Phycopomatus enigmaticus, hydroides, a couple of spionids, 3 of them, 5 psyllids, and 2 terebellids. And I'm sure there's more, because, as I said, the sampling has not been extensive. Most of the recent stuff has really concentrated on dock fouling. So it's ignoring what's in the soft bottom and in the various reaches. So probably everyone here knows this one, and quite well since it's so dominant. The Australian tubeworm, Phycopomatus enigmaticus. Very distinctive, very easy to spot, not just because it dominates, but because when you look at the tubes, they're conspicuously brown with flanges or rings that go around the tubes. If you can see the animal, this is what it looks like. And here's the operculum, which is basically a modified part of the crown that acts as a door stopper. So when the animal has completely withdrawn into its tube, this blocks the entrance to the tube, and most predators of the same or of the same size will not be able to get in. First discovered in 1921. This is a slide from Jim's PowerPoint from 2020 that he very generously shared with me. Another slide from his PowerPoint. It's just amazing how dense these growths can be. I've stopped at Lake Merritt through the years periodically, the Berkeley Marina and other places, and Petaluma, which is where I took this photo back around 2004, I believe, and this one as well. The growth can be 8 or 10 inches thick as generations after generations of worms settle out, grow, and die. And as Jim pointed out, it has a fascinating history of being transported around the world, that the early records, 1917, France, over here, and other areas were probably a result of boats during World War I. And then the populations that ended up here, like in San Francisco Bay, stayed in one place for a long time, and then suddenly they started showing up in other areas around the globe. And no one really knew why that was. And then the research in 2017 found that there are at least 3 genetic clades. And so one clade was the first one that went around the world during World War I. And then the other clades were transported at different times, and these were more adaptable and spread more easily. Jim, is that an adequate or reasonable summary?
That's great. That's right, Leslie.
Yeah. You did such a good job, and I'm sure that so many of the people that are here today were there for your talk as well. So really didn't feel the need to get into this one. There are probably 2 other serpulids, and serpulids are the fan worms that build calcareous tubes. The fan worms that build soft tubes are in family Sibellidae. So these are some nice photos that Cricket and Damon took. This one is probably I shouldn't say probably. It's likely to be Hydroides gracilis. I actually haven't seen any specimens, and neither of these photos show an intact operculum. Hydroides are easy to identify. In Lake Merritt, basically they have smooth tubes, but their operculums are double funnels. So there's a lower funnel and then an upper funnel. And the upper funnels, they have shapes that are species specific. So if I could see a photo of one where the operculum is clean and, showing the whole thing in its entirety, I could take it as species just from a photo. But unfortunately, none of the photos I've seen from Lake Merritt have shown it. Let's see. And I just want to compare that funnel with this one. This is very characteristic because it's covered with black spines. So even if you just see the animal, if it's got the operculum, it's very easy to identify. And here you've got the double funnel, the distal, which is usually white, and then the other funnel. This one is second only in iNaturalist observations to the Phycopomatus. This is family Nereidae, the ragworms or pileworms. And for years, well, ever since 1890s, it's been identified as first Nereia succinea. The name was changed to Alida succinea. At times it was called Neanthe succinea, and now it's settled on Alida, the genus Alida. Well, succinea was first described from Helgoland in Germany. And since it was first described, it's been reported pretty much all over the world. The problem is that all of those reports of succinea from places other than Germany were based on literature accounts that did not describe it very well, or else combined features of several different worms into one description. And none of those early reports, or even more recent reports, actually looked at specimens from Helgoland. So the German specimens were not fully characterized so that all of these records around the world could be compared to it. And when I first started looking at an iNaturalist, I was seeing photos by Damon in particular and some others. They were just different. Now, to understand how you can tell something is different when you're looking at a little tiny squirming worm, or even have it nicely preserved and laid out, you have to look at a lot of different characters. A Mexican researcher, Julio Villalobos Guerra, for his PhD thesis, looked at Neanthes and Alida succinea in particular, and other members of the genus. These are his photos of specimens from the type locality in Germany. And what I want you to notice are these appendages right here, which are called the tentacular cirri. They're short. All of them are short. In Damon's photos, there were some with short tentacular cirri, but there were some with long tentacular cirri. And some with sort of intermediates as well. These two are my own photos. So here's one with the long tentacular cirri. This one's intermediate, and this one is very short. Looking more closely at the specimens, and Damon actually sent me specimens that I could pass on to Tulio. Well, there are other differences in those as well. One of the other characters that we have to look at for these animals are the shape of the dorsal cirri, which are outgrowths of the parapodia, basically the legs of the animal. And in the posterior part of the body, they become very long. Now, these three on the top are from animals in Germany, and this one is from a San Francisco specimen. And they're different. And the shape of the parapodia, the other lobes, this is called the notopodial lobe. This is the neuropodial lobes. They have ligulus, they have accessory lobes. They're actually different. Here are some of Damon's photos, really quite nice. You can actually see the shape of the dorsal cirri here, and you can see how long the tentacular cirri are in that one. Well, so Tulio told me that there was another species in San Francisco Bay. At this point in time, he said we should still call everything Succinia because the morphological differences are not that great. In the old days, they would have been enough to separate them, unless you're a lumper, in which case you would just mush everything together and say this is natural variation. But there's a difference between intraspecific variation, which is the changes within one species, like, red hair, gray hair, blue eyes, brown eyes. And interspecific, which are the differences between species. But nowadays, you have to have genetic data to back it up. And genetic data so far has shown that populations around the world are actually a single species. But that's based on looking at just 2 genes. And Tulio is convinced that the San Francisco specimens, as well as specimens from other areas around the world, are indeed different species, but he's going to have to do entire genomes to prove it, to the satisfaction of where he's not going to be challenged by other researchers. So without going to DNA, we have to look at the morphological characters. And the ones that he noticed that are different include some of these: the length of the antenna, the length of the peristomial and tentacular cirri, the diameter of the eyes, the shape and length of the prostomium, which is this head region here. When they throw out their proboscis, you can think of that as a lining of the throat containing teeth, which, like an alien, can be thrown out to capture food and then bring it back into the body. We have structures, we have jaws, 1 pair of jaws, and then we have what's called paragnats, chitinous structures that help to hold on to their food. So the proboscis is divided up into zones, and how many paragnats do you have in each zone? How are they arranged? How much variation is them? So the shape, the number, the distribution, and how much bare space there is. Oh, and of course, number of teeth and the color of the jaws. For this group, the shape of the parapodia is really important. So you look at chetager 1 and 2, you look at anterior chetatures. These are some of the structures that you're looking at: the notopodial lobe, the neuropodial lobe, the accessory lobes, like this is called a ventral ligule. This is a median ligule. This is a dorsal ligule. We have dorsal cirri. We have ventral cirri. We look at all of these. We look at parapodia from the middle of the body, and we look at parapodia from the posterior. And I'd like to point out that these slides I'm showing you here are from a presentation that Tulio gave in 2015 to the Southern California Association of Marine Invertebrate Taxonomists, a group that for nearly 40 years has met once a month to discuss invertebrates and taxonomy and how to identify them. And the whole purpose of this group is to ensure that everybody working in the area, which was Southern California when we started, that we use the same name for the same species. It doesn't matter if the name is wrong. As long as we all use the same name when we realize a mistake, when new publications come out with a new name, a new assignment, we can all change at the same time. So our data is comparable for all the surveys that our members do throughout the entire area. So remembering that I had this, I asked him if I could use these slides to help explain what goes into distinguishing species. And for every family, every genus, whether it's psyllids, phalidocids, terebellids, spaghetti worms, hair worms, rag worms, there are all these characters that have to be very carefully considered and measured and compared. Polychaetes are called bristleworms. Polychaete actually means many hairs or many bristles. We have to look at those as well. And these are different types of the bristles that are found in the parapodia along the body. Some of the nerids, when they reach sexual maturity, transform themselves into swarming epitopes, and their parapodia can undergo drastic modification. So this is an animal that is not sexually mature. This is a parapodia of an animal that is sexually mature. It drops a lot of the regular chaetae, and it develops these specialized chaetae. We call them swimming chaetae, and they have paddle-shaped blades, and that helps them swim up to the surface to spawn. So as a result of his research for his PhD, in the Americas, he found these species. I took the names out because he is not published yet. But we have this species from the Argentina area, South America. This is East Coast. We have this species from California. These three species had been synonymized under Alida succinia. Julio was able to show very easily that they are not succinia because the differences are so pronounced between them. In addition, we have what he called new species 1 from San Francisco, species 2 from Connecticut, the US East Coast, and then one more from Veracruz. And since then, he has done more work and he has found more species. He's re-examined older species that have been synonymized, whether with succinia or some of the big European species like Branti and Virens, and he has both reinstated older names as valid species and described new ones. Unfortunately, once graduate students get their PhDs and go on to postdocs, they often don't have time to publish all of their research. So he still has not published this. He has a job working on invasive species, which takes all of his time. And as much as he would like to get back to this, he just has not yet. And I keep talking to him periodically and gently kicking him and asking him to do that. I was very surprised to see Nereis vexillosa on the list for Lake Merritt. Vexillosa is basically an outer coast full salinity species. It might show up in harbors, but only in areas that are close to the exchange with the open ocean. It likes full salinity. You're never going to find it in a brackish area which makes me wonder where it was found in Lake Merritt, if it was perhaps found near the area where seawater comes into the lake. Very easily mistaken for Aleta succinia. The differences that are immediately apparent if you know your polychaetes are the shape of the posterior parapodia and the arrangement of the perignath. So here's the true succinia again. And in this area, which is called zone 7 and 8, you can see the perignaths are fairly nicely lined up in 3 rows going across. In this area, 3, let's say that there's 25 perignaths here. Well, if we look at vexillosa, and this is a specimen from Monterey, you'll see that we have one strong line going across at the top of zone 7 and 8, and then you have a random scattering of them. So the arrangement is different, and here in 3, you have many more in a different arrangement. And then if we look at the posterior parapodia, here's the dorsal cirrus of a live vexillosa. Here's what they look like in life. And you see that this part is attached evenly to the tip of the elongated dorsal cirrus, which is long and strap-like. And here is true succinia. And you can see how the attachment is different and the whole shape is different. This one was a real surprise. I sent this photo to Tulio, and he agreed with me that it was probably Planedentata. This was, well, first off, I should say that this is one of the largest polychaetes on our coast. It gets up to 2 and even 3 feet long, with hundreds of the segments that give rise to the phylum name Annelida. In the past, this was synonymized with one of 2 European species. People sort of bounced it back and forth between Virens and Branti, which are now restricted in their distribution to Northern Europe and to basically Siberia. The reason why I couldn't take it to species just by looking at the photos is because the 2 species are very similar in appearance. And while Williami basically runs from Alaska through Washington, there are a couple records from the San Francisco area. And then Planedentata runs from about Bodega Bay down to San Diego. As far as we know, those are the records that are confirmed with museum specimens. So since they overlap in Central California, I couldn't just say that it was clearly one or the other based on distribution. It was very easy for me to see that this specimen that David photographed belonged to one of these 2 species was because of the shape of the dorsal cirrus, which is quite different from succinia. And here's the dorsal cirrus, which is attached back here, sort of at the base of this fat triangular blade, rather than at the tip, as in vexillosa and succinia. And here are the perignaths. Very different arrangement, far more perignaths than on either of the 2 species. Platynarius bicaniculata, which turns out to be a complex of, just on this coast, 5 or 6 different species, which is borne out by both genetics and by close examination of specimens up and down the coast. Has not been published yet. This one, to the casual eye, is easy to identify because it has the longest tentacular cirri of any of the ragworms, much longer than any of the other species that we have. So even if it's a bad photo, if it's from this coast, if it's an arid, if it has tentacular cirri that look like this, odds are pretty darn good that it's going to be in this particular complex. If you want to get involved in the smaller details, it has this very unique type of chaete, which you can sometimes see just with your naked eye or a magnifying glass. And this is what its paragnats look like, the chitinous structures on the aversible proboscis. Very different from the conical or round black paragnats in the Alida and Nereus. Another one of in the top 10, shall we call it, of observations for Lake Merritt is the Harmathoi imbricata complex. This is kind of a fun one. For most polychaetes, color patterns can be species specific. One species will have one color pattern, maybe with slight variation or intensity of color from dark to light. Well, in Northern Europe, where it's from, there are 10 different color patterns, and they all have the same genetic signature, so they're all one species. But then when we look at the genetics of specimens that have been barcoded for CO1, at the very least, and then the data is sent to the barcode of life database, there are 11 what they call bins, which represent genetic clades. So these are actually different species. And there are several of them on our coast. So for now, we're just calling it the complex. There are 4 color morphs that I've seen in everybody's photos on iNaturalist and my own collections. 2 are common, and I'll get to those in a minute. But I just wanted to show you one of the characters that's really important for identifying this as the Harmathoi imbricata complex. It has 2 pairs of eyes, and one pair is very obvious. The other pair is up at the top, sort of tucked away around the side. If you flip the animal over, you can see them quite clearly. So whenever anybody sees this in a member of genus Harmathoi, they automatically say imbricata. We have at least 6 species from the North Pacific that have this eye structure. So that's another something that needs to be looked at when considering just how many species we actually have here. Color patterns. There's this very dark one, which is quite common. I'd say at least half of the observations have this color pattern. There's this variegated pattern, which can go from dark and fairly solid to lighter, especially along the sides. And then we have ones that are red, but still mottled, solid red. And then we have this one, which matches one of the known European color patterns. Be really interesting to get the DNA for these different patterns in Lake Merritt. One other scale worm has been reported. It's Halicidna. I can't tell what it is from the photo because the 2 most common Halicidna species on the coast are quite variable. So both of these here are Brevicetosa, johnsonite tends to be orange. And their scales can be very smooth, like this, or they can be quite lumpy. And somebody's got to do DNA on these to see if we really have one species with a wide range of intraspecific variability, or if we have a number of species that are distinct genetically that overlap in their morphological characters. These three have shown up on lists. I have not seen any observations on iNat or in any of the samples over the years that I've looked at from Lake Merritt. These are small. They tend to live in the mud. It's really unlikely that people who are concentrating on the larger animals and on dock fouling would ever see them. This one, though, this one is photographed all the time. So along with Phycopomatus, Alida saxinia, Harmathoi, imbricata complex, Megacellus is right up there, the top four. Niponica. Nippon is another name for Japan. So this was brought here somehow from Japan. I first found this in Southern California in about 1997, 98. And on the first San Francisco surveys that Andy Cohen organized with Jim Carlton, John Chapman, Gretchen Lambert, and a number of other very knowledgeable, very wonderful people, I started seeing these in huge numbers. If I took a quart-sized jar of scrapings from a dock, I found thousands of these. And one thing that was really interesting to me is that in those early samples, there were ones that were bright green, ones that were bright orange, bright red, and bright yellow. Nowadays, the only ones I can find are these greenish yellow ones. All the other colors have disappeared. Were they different genetic species? Were there variants? I have absolutely no idea. It's family Psyllidae. It's very easy to identify by its very long, slender body. The long appendages that come out, the dorsal cirri from every segment. But easiest of all is the fact that the pharynx, which is an internal organ, which is irreversible, has a bright white and then a dark black band, which typically shows through the skin in live individuals. These reproduce by creating gametes, which all go into a posterior part of the body, which develops a pair of eyes and will break away and swim off. And this is the epitoke. So this is the reproduction portion that's going to swim up to the surface and spawn. And these get photographed a lot, as well as the adult worms. These are worms from down in my area. It's the same yellowish green coloration. We have the same nice white banding with the black afterwards. They're actually genetically different from the Japanese worms. And in the literature that I've seen for the Japanese worms, they're all green. So again, I keep wondering about the red, the bright yellow, and the orange ones that I saw in San Francisco in the early 2000s. This one is a gorgeous, gorgeous animal. It's another psyllid. Psyllids are united by the fact that they have a very distinct organ called a proventricle, which is basically like a pump. And these guys feed by throwing out their proboscis, and then the proventricle, the pump, pulsates, and it uses that as a suction device for picking up its food. This one was described in Australia, and over the years, there was a progression of sightings from different places around Australia, and then Japan, and then Hawaii. And then finally, in the late 1990s, I found the first one in LA Harbor. These guys, I believe, feed on yellow sponges and were probably transported in fouling on ship hulls, along with yellow sponge that they feed on. I mentioned that the megacillus, yes, the megacillus produces an epitoke. Well, these guys produce chains of stolons. So this guy is reproducing asexually by producing these stolons. The stolons, however, everyone in this chain will be either male or female. Everyone in this chain will be either male or female. So once these stolons break away, they will find each other, and external fertilization occurs. And the babies that grow up from that will once again reproduce asexually. So these are really fascinating animals and the variety of reproductive modes that they have. There was only one record of that from Lake Merritt. I'm a little surprised by that, but perhaps, Lake Merritt is just not a good place for the yellow sponge. Psyllus adamantia, another psyllid, distinctive color pattern of it's got these clear or unpigmented spaces in the middle of an otherwise light brown dorsum. This was described from San Francisco. What is it, an introduced species? Nobody knows, because it was described back, let's see, this was Treadwell, so 1914. We really don't have enough records back then to know if it was present before 1914. So it's thought of as a native, but I wonder a lot about this one, if we're going to find its relatives elsewhere in the world.
Leslie?
Yeah.
Can we take a pause here for a couple of questions that have come up, and then some quick with some quick closing slides, and then reopen for more discussion? Is that something that you could do?
Sure.
Okay.
Yeah.
So we had some great questions here. And I think since we were just talking about it, we had a question from David Wofford. He wondered if the different color morphs of the megacillus had disappeared.
I haven't seen them again.
So, wow.
Yeah.
Are they fixed traits, or are they plastic? Do they change in different different habitats and whatever?
Well, when I saw the first ones, I thought perhaps that was a result of feeding, because polychaetes can have 3 different types of color. One is the color may reflect what they're feeding on. So if it feeds on red algae, it'll be red. If it feeds on yellow sponge, it will be yellow. It can have external pigmentation, like in the Psyllus adamantius, which has got a brown body with those clear spots in the middle of each segment. And there can be subdermal color, which can be like black spots that are actually on the digestive tract, which show through the body. When I first saw those 4 different colors of the megacilus, I thought maybe they were feeding on different things. I have absolutely no idea.
That's fascinating. And we had another question, which um, from Mario, and he wondered if you could tell us a little bit in details about how to prepare polychaetes for microscopic identification and how to take these high quality images that you have.
Ah,
I bet there's a long answer to that.
yeah, actually there is. That's a real early one. So that's Damon's. These are mine. To prepare them, well, it depends what I want them for. If I want them for morphology, I will put them in formalin first. And after a few days, rinse them in fresh water to get rid of excess formalin and put them into alcohol. If I want them for DNA, they go straight into 95% alcohol. Usually what I'll do, though, is save most of the body in formalin and just take off a piece of tissue and put it in the 95 for DNA. That way I can do both the morphology and the DNA. For the photography, boy, I went through so many iterations of trying to learn this stuff. Okay, I photograph live animals here. So these have been narcotized with magnesium chloride mixed with fresh water. And this prevents the transmission of signals through the neurons. So the animal can't move. It's basically paralyzed, which allows me to clean it and pose it. I will either photograph them on a glass dish, which is suspended above a black background. And it's suspended so that whatever is going on in the background is blurred out and it's a solid black. Most backgrounds, however, in photography, using 2 strobes coming in at an angle or one at an angle and one like this to bring out the characters, most backgrounds, no matter how black they look to your eye, are going to come out gray in the photograph. The absolute best photographic dish to photograph these things in are, wait for it. If you can see this, this is a Las Vegas black glass ashtray. These were extremely common at one time. This is the blackest glass I've ever seen. And any photograph you take of an animal sitting on this dish will have a solid black background. Can get them on eBay. Make sure they're not scratched on the bottom because the scratches will show in the photographs or reflect light. And they're cheap. Absolute best. And one other thing, filter your water or your magnesium chloride or whatever you're shooting in. If you have particles in the water, they will reflect light and that will affect the resolution. So that's it. The more care you take preparing the animal and prepping everything, the better your photo will be.
Well, these are beautiful, and thank you so much. Let's see, I have one new message here. Oh, are there any polychaetes that we should be looking for in Lake Merritt aside from the Etione and the with good pictures of operculums and hydroides? Any other ones we should be on the lookout for?
All of them.
Okay.
Take specimens. And actually, I was chatting with Tulio just today, and I mentioned that I would be presenting this talk, and he would love to get specimens of Nereids so that he continued to work on this question of who exactly do we have here in San Francisco? Do we have Succinia? Do we have one undescribed species? Do we have several undescribed species? He's going to be working with a researcher in Taiwan who's going to do the whole genome workups. And the more material he can get from this area, your area, not just Lake Merritt, but the whole San Francisco system, preserved in 95%. Everclear is good for that as well, if you can't get true 95. You know, the more evidence that they will have for confirming presence and absence.
Wow.
So he would love stuff to be collected, and then that material can be sent to me, and I can give it to him next time I see him.
Great. That's a great question too. Well, I'd like to take just a pause, and if Leslie's able to stay after this, our customary thank you slides at the end, we'll reopen if you're able to stay for a little bit longer after that.
Yeah, there are more animals. I could just go through them, you know, like 3 seconds each, just to show you the variety that we've seen in Lake Merritt. Because,
wow.
You know, this is what I've gone through 10 so far. And
that sounds wonderful.
You know, only for those people who are willing to stay.
Okay, so we're going to pause right now in this wonderful presentation. We want to thank Leslie, of course, and many people who have come to Lake Merritt to look at the biodiversity, test the water, and do stewardship that are listed here on the slide. It's really been a community effort. Coming up next, I want to invite everybody to come to our December 6th program on Friday night, 7 p.m., free on Zoom, when Ben Chen, who is one of the senior workers at the Lake Merritt Water Quality Management Pilot Project, Lake Tech, will be talking to us about the oxygenation infrastructure and progress and what's going on out there with all those things bubbling and in the lake, what are some of the insights that they have and so forth. We are planning to have a stewardship workday on the third Saturday, which will be coming up on November 16th. And details about this will be coming out on our Facebook site. Okay, our Lakeside chats are recorded and rebroadcast by the City of Oakland KTOP Channel 10 TV. And they show a collection of old shows. We have now up to 48, so we've got a lot of variety there in the first 2 weeks of the month, and then in the second 2 weeks they will show the current presentation. So if you enjoy seeing what we do, please consider making a donation. We have a lot of all of our staff are volunteers, but we do have a lot of consumables and equipment that wears out, needs replacing, in order to serve our young people and just the general community when they come out to see our work at Lake Merritt. So you can do that on our website. We have a button which goes to PayPal, or you can also mail a check to Rotary Nature Center Friends at the address shown here. This is what our website looks like, so you can locate the button. We'd like to thank some of our long-term supporters, in particular the Frederick E. Hart Foundation for Educational Opportunity, for donations every year that help make our Lakeside chats possible. I'd like to thank all of our volunteers, program participants, and partners that allow us to do our work at Lake Merritt. We are primary stewards of the Lake Merritt Wildlife Refuge, and we host groups to the lake for bird watching, nature walks, native plant identification, planting and weeding, and water testing. And so we are stewards and interpreters and supporters of all of the good work that goes on at Lake Merritt to help people and nature connect in a productive way in the heart of the inner city. So thank you for coming and joining us tonight. So now really are very happy that we can continue our conversation. And let's see. Oh, there's some more questions here. So Valerie says she has never searched for polychaetes before, but now she really wants to do that. What equipment do you need, and what places other than Lake Merritt can you find them, the polychaetes? I would like to say we always focus on the dock because it's easy, but what other things, what other places and other tools and things would you suggest to us for following that?
Okay, so polychaetes are everywhere in the ocean, from the beach on down to the deepest areas. They're in mud, they're on rocks, they're in kelp holdfasts, among seaweed, among bryozoans, dock fouling. They're everywhere. So for dock fouling, you want a scraper. You want a pan to spread things out. Some tweezers or a pipette to get them out. A shovel, dig up some mud. If you've got a boat and you've got a grab, you can do that. Just get a bucket. To photograph them, don't just put them on your hand, because the water tension will make all their appendages cling to the body and to your hand. So they don't look normal. The appendages are not in normal position. And I see those photos in iNat and I just hate them. When I go to the beach, to a dock, I always carry around an assortment of little trays, little pans. I fill them with water and I put the worms inside, so all of their appendages are in normal position. Same as if you were doing a nudibranch. You don't want it to be a blob out of the water. You want all its serrata and everything standing up in its full glory. Yeah, it's a pretty easy way to start. A magnifying glass is good for the smaller stuff. And basically, as you sort through your sample, you're looking for movement. You're looking for flashes of color. You're looking for body shape. With time, you develop an eye. I think that just by going onto iNat and looking at the photos others have taken of, good-looking worms in their natural habitat, you can see what you want to look at. Go look at a sponge. Go look at that, fluffy gray stuff on docks. That's often tubes of polychaetes like spionids or amphipods, and there'll be polychaetes crawling around in there. The sponges, the seaweeds. Yeah. I remember all those colorful megacilus from my first San Francisco collections. They were among mostly seaweeds and sponges and bryozoans.
We certainly have a lot of that very close at hand at the docks at Lake Merritt. We sometimes bring out a dissecting scope, which can give you a closer view, but photographing in the field is kind of hard.
Yeah, I'd like to take, if I'm not going to photograph the animals, in their native habitat, as so many people do, especially with docks and intertidal, I like to take them back to my lab where I can get them out of their tubes. I can use a magnesium chloride to relax them, brush their hair, wash their faces, and make them look pretty for their close-ups.
I had no idea. Yes, more questions. Let's see. Well, given us so much to look at. It says, someone is saying that it helps to dissect, to ID the posterior sections of polychaetes.
Yeah. When you're looking at the narids and you want to look at the posterior sections, magnifying glass is good. Once you develop an eye for it, you can look at them in the field and know what they are. Because of the way they look, the way they move, their coloration. It's just a matter of developing the eye. And again, iNaturalist is wonderful for that. I mean, that's why I started on iNaturalist, so I could see more photos of live animals and get a feel for what they look at. Where do they live? Are they in tubes? Are they in seaweed? Do they have a characteristic shape? So iNat, it's wonderful.
And yeah, it's and so easy to use, and you get better with time sometimes.
Yeah, definitely.
We have any more questions? Please raise your hand. And this has been a fabulous view of all of photos. You have more photos to show?
Yeah, I can go through there quickly, and
and these are all up on iNaturalist, right?
Mine aren't.
Oh, they aren't. Oh, okay.
Okay, so Sphericillus, Amblyocylus, nigrolineata from Japan. This is Marphysa, reported in the past as Californica, Sanguinea. It's probably Californica or an undescribed species. All that flock you see on the dock, a lot of it is made by spionids. Polydora canuta is one. So this is where you would scrape it off and you would stir up the flock. And often the worms will swim out of their tubes, theoretically to safety. Here's another view of the tentacles of the spionids sticking out of their tubes on the dock. And if you do plankton samples, you're going to see tons of baby spionids. Capitellets, these show up. These would be more common in the mud at the bottom. Ceratulids, these are called hairworms, and they're a total pain to work with. But if you see these colorful filaments coming out of the mud or between mussels, probably a ceratulid. There are at least 3 species there. Terebellids, spaghetti worms. Neoamphitrite, I found this for the first time on one of the rapid assessment surveys with Andy and Jim. This is a rope at Pete's Landing in San Francisco Bay, which is completely covered with the tubes of Neoamphitrite. It's introduced. It's sporadically invasive, and it's undescribed. But it's amazing, and in some areas it absolutely dominates both docks and the mud bottoms. And here are pictures of the live one showing some of the features that you have to look at in order to identify them. Another one, Nicolea. This one is actually an invasive down in Southern California. So I was really surprised to see that a few of them have finally made it up here. Neoamphitrite has not made it down to Southern California yet. Then we've got the fan worms. This is a native. Here's another one. This is actually from the boathouse. This one is from the Sea of Japan. So this is an introduced species. I don't know how long it's been here. I have some old museum samples from about the 1940s. It may possibly be this species, but they're in bad shape. So I can't tell for sure. This one has shown up by Spira pacifica. I'm very surprised because it's a full salinity worm, but very characteristic look of these alternating rows of purple spots. Very, very pretty. Myxipula, the slime worm. Big question as to whether they're introduced or native. Could be both. Branchioma. This one is definitely introduced. This one is very definitely an invasive. In San Diego, where it's common, it completely covers large areas of the docks and pilings. Very easy to tell because on the outside of the feathery crown, you see these little filaments on the outside. None of the other sabellids have filaments on the outsides of their feathery crowns. And this one's tiny. This is just a couple of millimeters, very simple body. They're identified on the basis of their jaws. And I think the ones that I found in your sample, Katie, are the first reported for Lake Merritt.
So are they do they live in tubes or are they just no? What do they eat with those jaws?
They're raspers. So they're rasping off diatoms and microbial films from the surfaces of mussels or rocks or whatever. So they use their jaws to rasp off their food. And they're very tiny. Some do build tubes, some do not. And that's pretty much it. And none of these are in Lake Merritt, but they're really pretty. And just thank you all for being for willing to listen to this and put up with my obsessiveness about polychaetes.
Well, I think it's really made a number of us want to get out there and take a closer look than we may have done in the past. And that's amazing. There's so many of those in the and we have somebody has raised their hand. James Urban would like to ask a question.
Sure.
See,
Jim, are you there?
I guess if you can hear me now. Can you hear me? Yes.
Yes.
Yeah. Hi, Leslie. Yeah, really interesting. Yeah, I work in Lower South Bay, and I know absolutely nothing about polychaetes except that we catch them once in a while. We catch the Alida succinea, and I'm just kind of curious. All of ours have the long tentacular cirri. I photograph them often, and I'm really fascinated with them because they have the eyes and the long tentacles, and they seem to be a very intelligent worm. Have you ever noticed that they interact? They fight with each other very frequently.
Yeah.
Yeah, they're and what's up with that? They are sexual, is what I understand. I mean, they can be kind of hermaphrodite or whatever, but they do have sexes, and I don't quite understand. At one point I thought they were universally hostile towards each other unless they get into the epitopic phase, but then I found out that some of them don't. So what's up with that?
Okay, so when you see them fighting, is this after you've captured them and you've got them in a dish?
Yeah, in a photarium, and they're kind of like moving around in the photarium, and they'll encounter each other. And I mean, I've had them literally face off against each other and bite each other, and they use the palps. Apparently, they have a little bit of venom in the palps, maybe not much, but I mean, you can tell when one bites the other, it must hurt.
Yeah, a big nerd can put you in a world of hurt. Well, okay, there are really two factors there. One is the fact that they are territorial and, get off my turf. But the other is that you have just taken them from their homes and thrown them, in into this container with all of these other worms. And so they're very upset. And so they're just, going to be fighting. At the same time, these worms are what we call thigmotaxic, which is that they need the feel of something along their bodies because they live in tubes, they burrow through the mud. And when you put them into a container with nothing but water, first they'll head for the walls and, stay along the walls. The other is that when they meet each other, they may just start coiling around each other, and eventually turn into this giant knotted ball. So when I catch a lot of worms, I will separate them into different containers just so that they don't end up in a horrible tangle that I cannot pull apart without damaging them.
Yeah, the other, that makes a lot of sense. The other thing I've noticed too is only on a few, well, no, on every occasion, if I put them in a photarium, which is essentially a cubic shaped, aquarium, a tiny aquarium, they're the only critter amongst all the mollusks and all the fishes that can actually figure out how to escape from a photarium. They crawl around for a few minutes. They figure out it's a square, and they'll go up and out of the water, literally out of the water, and climb out like an octopus and try to get out of that enclosure.
Oh God, I have all these photos of worms crawling out of my dishes. Narids, always narids that are crawling out of the dishes and off the table, and you find them a couple of days later dried on the floor somewhere.
Yeah, I call them the genius worms. They're geniuses. Yeah.
I don't know if they're
okay. Yeah.
but when they're out of their tubes, or even when they're when they're out of their tubes, they're moving all the time. And so that's just a continuation of their behavior.
Yeah. Yeah, thanks. Thanks. That was very helpful.
Uh-huh. Thank you so much. Well, it's getting late, and we want to thank you so much for staying and talking and giving us this wonderful picture of what we might have in our lake and just what might be out there in the world. Are there more questions? I don't want to miss anybody, but
yeah,
if not, thank you so much.
One last thing, Katie, that anybody would like to collect narids for research, you can contact me through iNaturalist. I'm Leslie H.
Okay. Yes, that sounds wonderful. I will put that in our post chat email that that they can do that, and that that'll be great. And ways to contact you and some of the other references that have been thrown out, any other ones that you might want to share with us, that would be great. Okay. Well, have a wonderful evening, and thank you so much. Thank everybody for coming tonight, and please mark your calendar for December 6th. We'll hear about water quality and what's going on in the lake.
Thank you, Leslie. Thank you so much. Thank you, Katie.
Thank you very much.
Yeah, thank you to everybody for joining us tonight. This was really lovely.
Yeah, thank you for having me.
It was such a pleasure.
To a bunch of newbies and convert them to the love of worms.
Yeah.
Absolutely.
Okay. Okay, everyone.
Your obsession is catchy.
Thank you. Okay. Bye-bye.
Okay. Bye, Leslie. And again, thank you so much. It's wonderful.